Hello everyone, and welcome to this screencast about in silico PCR in the UCSC Genome Browser. In this video, we will demonstrate how to use the UCSC Genome Browser's in silico PCR tool for predicting the success of PCR experiments in molecular biology. PCR, which as you know stands for polymerase chain reaction, allows multiple copies of a particular fragment of DNA to be synthesized. PCR is an important and ubiquitous tool in molecular biology that has many uses, including medical diagnostics, forensic DNA analysis, cloning, and genome analysis. By the end of this video, you should be able to explain the utility of in silico experiments versus in vitro and in vivo experiments, navigate the in silico PCR tool in the UCSC Genome Browser, and perform in silico PCR to determine any potential off-target PCR products in your experiment. I'm going to start by explaining the utility of in silico experiments. Biological sciences aim to increase the understanding of biological processes by experimentation, and one way of classifying experiments is by the environment in which they are performed. These can be in vivo, in vitro, and now in silico. In vivo is a Latin term meaning within the living, and in vivo experiments refer to those that are performed in a living organism. For example, experiments performed in mice or clinical trials performed in human subjects. In vitro literally means in glass and refers to experiments conducted in vessels such as test tubes or petri dishes. In vitro experiments involve studies on cells or biomolecules that are performed in a controlled environment but occur outside of their normal biological context. In the 1980s, the term in silico was coined to refer to experiments performed by means of computer modeling or simulation. In silico literally means in silicon, referring to the fact that most computer chips are made of silicon. A major distinguishing factor between in silico experiments compared to in vivo and in vitro experiments is the time scale on which they occur. In vivo and in vitro experiments occur on natural biological timescales that are limited by the physical and chemical properties of the biological components involved. However, in silico experiments simulate biological processes on timescales that are many times faster than reality. The speed of these experiments are therefore only limited by the processing power of the hardware and the efficiency of the algorithm. Traditional molecular biology experiments occur in vitro, and PCR is no exception. But testing and optimizing the design of an in vitro experiment can oftentimes be time-consuming and very costly. The most basic PCR experiment requires designing and synthesizing primers, extracting DNA from a biological sample, assembling the PCR reaction, carrying out the reaction, and then analyzing the results. This whole process can often take several days. But using online databases that house genomic data, such as the UCSC Genome Browser, we can make use of computer simulations or in silico experiments to predict the outcome of a PCR reaction in a matter of minutes. The parallel process for an in silico PCR experiment is as follows. Design the primers as you would have for an in vitro experiment. Then identify a target genome sequence data. This often takes minutes by browsing through a database of online genome sequencing. Carry out the reaction, which can take seconds once you set the algorithm running. And then finally analyze the results in a matter of minutes. A successful in silico PCR experiment on the most complete genome of interest is a good sign that your in vitro PCR will not contain any off-target products. Now that we've explained the motivation for in silico experiments, we're going to demonstrate the UCSC Genome Browser's in silico PCR tool. Before we go any further, I'd like to make sure that you're comfortable with how PCR works. In the next section, I will assume you're familiar with the following: stages of PCR process, primer design, 5' to 3' directionality of DNA polymerase-mediated strand elongation, and PCR products or amplicons. If you're not, please pause this video and go to the website on your screen for a refresher. Recall that a successful PCR experiment is dependent on primers that are efficient and selective. The UCSC Genome Browser's in silico PCR tool works by searching a sequence database or a particular genome sequence with a pair of PCR primers, and then using the results of this search to return the possible PCR amplicons or all sequences in the database that lie between and include the primer pair from such an experiment. To access the tool, go to the following website and then click on Tools and then In Silico PCR. So now I'm going to go to a web browser and show you how this tool works. So we go to the following website, genome.ucsc.edu, and we click on Tools or hover over Tools, and you can scroll down until you get to In Silico PCR and click on that. As you can see, the tool looks pretty simple. There's not that much going on here. So I'm going to walk you through what each one of these parameters means. The first parameter is the genome. So in this case, I'm going to work with a human genome. But there are many other organisms that you can browse the genome of. The second stage is the assembly. So it's a good idea to always use the most up-to-date version of the genome that you would be performing your in silico experiment on. That's because that most up-to-date version is oftentimes the most complete. So in this case, for the human genome, the most up-to-date version listed here is from December 2013. The next parameter is the target. So for this particular set of selections that we have made so far, we can either use the genome assembly as the target or GenCode genes. But in this case, I want to use the genome assembly because the PCR experiment I'm going to perform is going to involve genomic DNA that has been isolated from a human person. The next two boxes are where you would input your forward and reverse primers, and these would have been primers that you designed for your in vitro experiment. This box, max product size, indicates the largest PCR product that you would like the program to return. So it may be possible for larger products to be to occur from your PCR product, but you won't be shown those results if it's larger than this number. The minimum perfect match of 15 is the number of bases that match exactly on the 3' end of primers, and in most cases the minimum match size is 15 because on the 3' end of primers you need to have perfect matching in order for a reaction to proceed in real life. And then the minimum good match is the number of bases on the 3' end of primers where at least 2 out of 3 bases match, and these are just default settings which we're going to move forward with. Finally, this flip reverse primer button that you can toggle on and off allows you to change the directionality of the primer that you put in the reverse primer box. So remember that primers are 5' to 3' in direction, and depending on how you've written that primer out in this box, you might need to flip the direction in order for the PCR reaction to occur. All of the settings for the in silico PCR experiments are explained right here on this page. So now we're ready to do a worked example. I'd like to do an experiment to amplify part of a human gene for a diagnostic test. I've previously designed some primers down here that are going to amplify the region of interest of the gene, but I want to use an in silico PCR to check that my primers are selective and will not produce any other off-target PCR products. So this is how we're going to go about doing this. I've selected my genome as human. My assembly is the most up-to-date assembly from December 2013. My target as a genome assembly rather than the GenCode genes. I'm then going to copy and paste my primers into the forward and reverse primer box. Now I want you to note that the forward primer and the reverse primer that I have listed here are listed in the 5' to 3' direction. So for the forward primer, that's fine. I'm going to copy that and then I'm going to paste it in this forward primer box. For the reverse primer, it's listed in the 5' to 3' direction, so I'm going to copy it and then paste it in this box. But remember, I'm going to have to flip that reverse primer because it's going to bind on the complementary DNA strand and be in the opposite direction. So once I've entered all of this information, all I have to do is click Submit. And there you have it. This program has essentially run an in silico PCR experiment where it's used these two primers to find all possible amplicons that are less than 4,000 base pairs in size in the current build of the human genome. And the results are produced here. So this pair of primers actually shows that 2 amplicons were produced. This is one amplicon. It's on chromosome 10 from this position to this position, and it's 316 base pairs in size. The program actually shows another amplicon on chromosome 19 from this position to this position, and it's 714 base pairs in size. So let me just walk you through what these results actually show. Here we have the forward primer, and here you have the reverse primer. I want you to note that the reverse primer as listed here is actually the reverse complement of the primer that we have listed here. So if we were to read the reverse complement of this primer, we would start on this end and it would be T, C, A, G, etc. And that's listed as you see up here. So we got the directionality correct, and that's how we were able to produce this product. And over here we have the primer and then the sequence that is listed in the reference genome as occurring in between the 2 primer pair sequences. So we see that the result of this experiment is that using these 2 primers, we were actually generating 2 different PCR products in very different parts of the genome. Now, if I were to go about doing an in vitro experiment using those 2 primers, what would happen is I would get a mix of products, and my reaction is not going to be specific to the product that I was interested in obtaining. So the next step from here would be for me to go back and redesign my primers so that I don't have this problem of generating multiple amplicons, especially multiple unwanted amplicons, I should say. So when your in silico PCR shows a successful result of only showing 1 amplicon, that's a good indication that your in vitro PCR experiment is going to be successful. So, in summary, in this video, we discussed how in silico PCR experiments can efficiently simulate in vitro and in vivo experiments. We demonstrated the UCSC Genome Browser's in silico PCR tool. And then finally, we illustrated how in silico PCR can help facilitate successful in vitro PCR by predicting off-target results. I hope you found this video useful for furthering your understanding of PCR design, and I encourage you to try out the UCSC Genome Browser's in silico PCR tool when designing your own PCR experiments in the future.